Presenting you Meditainment. Experience the joy of learning medicine.
In the previous video, mutation at the level of the receptor was well described. In this video, we'll be seeing about the mutation at the level of the adapter molecule RAS and mutation at the level of the signal transduction pathways, the PI3K pathway and the RAF pathway. Mutation at the level of the adapter molecule RAS. Gain of function mutations in the RAS adapter molecule is the most common aspect seen in most of the tumors. Like in 90% of pancreatic adenocarcinoma and cholangiocarcinoma, and 50% of colon, endometrial and thyroid carcinoma, and 30% of lung adenocarcinoma and myeloid leukemia, we have this RAS adapter molecule mutation. Now let's see who is this RAS and how does he get the role of the villain. So the RAS protein belongs to the family of membrane-associated G protein. When the GTP is bound to the RAS, he is in the activated state, and when GDP is bound, he is in the inactivated state. So now let's recall the receptor tyrosine kinase pathway. Initially, when a growth factor binds to the receptor, there occurs dimerization of the receptor and the tyrosine residues, they get phosphorylated. And then the adapter molecule, the RAS, it comes and binds to the receptor. Initially, the RAS is in the inactivated state. Later on, it gets activated and then it in turn activates the other two pathways, PI3K and the RAF pathway. As we know before, once the process is over, the activated RAS turns back into the inactivated RAS and the cascade comes to a halt and eventually cell proliferation stops. But how does this happen? Yes, RAS intrinsically has an GTPase activating protein called the GAPS. This protein activates GTPase, which hydrolyzes GTP to GDP, thus making active RAS to the inactive form. This inactivated RAS can no longer activate the other pathways. Therefore, cell proliferation comes to a halt. What happens in several distinct RAS point mutations is that this GAPS activity is reduced. So this leads to trapping of the RAS protein in an activated state. This all-time euphoric RAS, due to no GTP hydrolysis by GTPase, it keeps activating the pathways, which leads to more cell proliferation and finally leads to tumor. So it can be said that the consequences of gain of function mutations in RAS proteins should be mimicked by the loss of function of GAPS. In familial neurofibromatosis type 1 tumor, there is a mutation in the NF1 gene. This NF1 gene, in general, it encodes a GTPase activating protein. Now, when there is a mutation in the NF1 gene, it leads to the production of less efficient GTPase activating protein, which causes the RAS to be active all the time. So here, the NF1 acts as a guardian angel. So NF1 gene can be called as a tumor suppressor gene since it acts through a negative regulation of RAS signaling, thereby suppressing danger tumor signals. A point to note is that mutation in the tumor suppressor gene is one among the 8 hallmarks of cancer. So this will be dealt later in the further videos. Since RAS mutations are more common, targeted therapies specific for this protein were developed. But unfortunately, strategies designed to target RAS has so far not been so successful because the RAS protein keeps changing its structure and its mode of signaling, which makes it a difficult protein to inhibit with drugs. So now let's move on to the mutation at the level of the signal transduction pathways, the PI3K and the RAF pathway. As we have mentioned before, the RAS adapter molecule activates the RAF pathway and the PI3K pathway. Gain of function mutations in these pathways leads to various tumors, which means that these pathways get activated without any trigger. Mutations in the RAF pathway are seen in 100% hairy cell leukemia, more than 60% of melanomas, and 80% of benign nevi. Whereas mutations in the PI3K pathway is very common in certain cancers. In 30% of breast tumors, this
As
mutation
in
is
an
seen.
all-time activating RAF pathway, there is a serine threonine protein kinase called BRAF. This sits at the top of a cascade of other serine or threonine kinases of the MAPK family. Like activating RAS mutations, activating mutations in BRAF stimulate each of these proteins in the MAPK family and ultimately activate the transcription factors, leading to rapid cell growth. On the other hand, the all-time activating PI3K pathway activates a cascade of serine or threonine kinase proteins like AKT. This AKT has three functions. The first function is it activates mTOR, which is a sensor of cellular nutrient status. This leads to stimulation of protein and lipid synthesis. The second function is it inhibits a protein called BAD. This BAD is a pro-apoptotic protein, hence no apoptosis takes place. The third function is it negatively regulates or decreases the production of FOXO transcription factors. These FOXO transcription factors help in turning on the genes which promote apoptosis. As this is negatively regulated, no apoptosis takes place. These three functions finally lead to rapid cell growth. A point to note is that this PI3K pathway is being negatively regulated by a protein called PTEN. This PTEN is a tumor suppressor gene protein.
BRAF inhibitors in advanced melanomas have shown striking clinical response. These many studies about the specific oncogene and their resulting tumors highlights the need for molecular analysis of tumors to guide appropriate therapy, a phenomenon termed as oncogene addiction. That is, the tumor cells are highly dependent on the activity of an oncogene. Hence, finding the oncogene is very essential in treating a tumor. The rest will be continued in part 3. So the left out part, or the mutations at the level of the transcription factor, mutation at the level of cyclins, and mutation in non-receptor tyrosine kinase.